Hello and welcome to this 44th session of the International IVF Initiative entitled Reboot, Can Cytoplasmic Errors Be Averted or Remedied? This session is kindly co-sponsored by both Embryo Tools from Barcelona in Spain and Nadia Clinic in Kiev in the Ukraine. So thank you very much for your sponsorship. My name is Jacques Cohen and I'm one of the founders of I3, together with Thomas Elliott, Peter Nash, Shaister Sadruddin, all from Atlanta, Georgia, Colin Howells from Geneva, Switzerland, Marianne Servetic from New Jersey, and our Executive Director from Cardiff, Wales, Charles Palmer. I am located just south of Hudson, New York. You can look it up on the map. It's about 2 hours north of the city of New York, and it's on the river. And I have the great pleasure and honor of introducing you to four of my really best friends. They're all specialists in the topic of altering ooplasm for clinical applications. Professor Eli Adashi from Providence, Rhode Island, Brown University, who will speak about the very exciting topic of either replacing or genetically repairing mutated mitochondria in eggs from women at risk of transmitting mitochondrial disease. The three other speakers, on the other hand, will cover the controversial yet equally exciting topic of using cytoplasmic replacement derived from donor egg cytoplasm for treating patients, rare patients, who have failed IVF multiple times. They are Dr. Serena Chen from St. Barnabas Medical Center, New Jersey. Hi, Serena. Serena will also be my co-moderator. Dr. Nuno Costa-Borges from Barcelona, Spain. Embryo Tools, and that's his QA/QC team. And finally, Pavlo Mazur from the Nadia Clinic in the Ukraine. Both Pavlo and Nuno are embryologists. Doctors Chen and Adashi are everything. They are, of course, medical doctors first and foremost. So all speakers today will be kind of adjunct moderators and panelists because they are talking about the same content, and they are welcome to ask each other questions or extend to the other lectures. So we have a great program planned for you for the next 90 minutes. Could be a little less, could be a little longer. Please remember to ask questions, and you do that by clicking on your Zoom window and then going to the Q&A portion on the bottom. And that's where you ask your questions. The chat room is really for chatting. It's just for talking to each other. So we don't look at that portion to find questions. Now, both Serena and I are assisted by a wonderful triage team who will check the questions and transfer those to a separate document and present that to us. So don't worry if there's a lot of questions, and we really invite you to ask as much as you can. So, but first let me introduce you to Dr. Serena Chen. She's my co-moderator and also the second speaker. I've had the honor to work with Serena while at St. Barnabas Medical Center in New Jersey. Serena is a reproductive endocrinology and infertility specialist. In the United States, we call this an REI. And she's also the medical director of the Institute of Reproductive Medicine and Science at St. Barnabas in Livingston, New Jersey. I used to work there in the last century. She's the clinical associate professor at both Rutgers medical schools. There are two of them. She's a professor there. And Dr. Chen is a person who always strikes me as incredibly passionate of reproductive science. She's a fan of reproductive science. She is amazed and grateful each day at the explosion in reproductive technology. She will email, text me, call me about papers and ask questions or make comments. So Serena, it's a pleasure to have you with us today, and looking forward to this session with you and your introduction of Professor Adashi.
Thank you so much, Dr. Jacques Cohen. Our first speaker, I'm very honored to introduce Professor Eli Adashi. Dr. Adashi is a really remarkable person. Has had 3 phenomenal careers in medicine and science, and just any one of which would put him kind of at the top in terms of global leaders in reproductive medicine, but he's had three, over 600 publications as a reproductive endocrinologist, an REI. Uh, um, and and that in itself is a significant achievement. He is the dean of the Brown University Medical School, a place very close to my heart. And he now is a prolific author and global thought leader in reproductive medicine, specifically in the legal, ethical, medical, and scientific aspects of genetic engineering, germline gene modification, and the implications of this incredibly exciting technology for the future of the human race. And I love reading his thoughts. If you haven't read all his articles, I have them all printed out here. They're just some great stuff. Great reading. He's published in numerous scientific journals, but also has been recognized in the public by places like the New York Times and the Washington Post. I'm very privileged to have known Dr. Adashi. I'm not sure he remembers this, since 1994, when I became a fellow, or 1992. No, I've known you since 1992. 1992, when I became a fellow at the Johns Hopkins Hospital, Reproductive Endocrinology. And Dr. Adashi, you were at the University of Maryland. And every time I drove Dr. Wallach, my chairman at Hopkins, crazy with my research project, and I got stuck, he would call up Dr. Adashi and say, "Can you help Dr. Chen? Because she seems to be struggling." And his insights and mentorship meant so much to me. Eventually, I did pass my boards. Dr. Adashi was one of my examiners. He was very tough on me, but it was still a good experience. And anyway, I'm just so honored to be here with Professor Adashi, who's won numerous accolades recognized by numerous organizations all over the globe, including the National Academy of Science and Medicine in the United States, the Royal Society of Medicine in the UK, as well as other analogous bodies, scientific bodies in other nations, Poland, Canada, China, and France. So without further ado, Professor Adashi.
Thank you very much, Serena. And yes, I do remember you from way back in the 90s. It does seem like a century ago, which it is. But these were good times also in many ways. And Lord knows, there were no pandemics. And that alone made a huge difference, except we didn't realize it, of course, at the time. But thank you for the very kind introduction. My role today will be to provide somewhat of a background to where we stand with the germline remediation of mitochondrial DNA diseases. As you know, these are very serious and at this point untreatable conditions. And to try to do justice to the entire field, what I was going to do is read essentially a very brief statement without slides, do it relatively slowly, and hopefully in so doing, give you an overall view of where that field stands today. Matrilineal transmission of mutant mitochondrial alleles, as I've just mentioned, gives rise to inborn metabolic maladies that feature different penetrants, present differently, and come along with divergent prognosis. Undergirding this spectrum of multisystem disorders are various states of what we call heteroplasmy, wherein mutant mitochondrial alleles coexist with wild-type counterparts. In the United States alone, we estimate about 1,000 children are born every year who are affected with mitochondrial DNA disease. All of those are beyond the reach of contemporary medicine. We have very little to offer other than supportive, palliative therapy. Effective mitigation, let alone cure, really remain distant goals at this time. Recent scientific inroads, however, give rise to hope that this relatively grim state of affairs might be alleviated via one of two distinct paths of germline remediation, meaning that you will have to undergo IVF, of course, to benefit from these technologies. The first, or in the first, mitochondria of oocytes or zygotes at risk could be replaced with donor unaffected counterparts. This is what we call mitochondrial replacement therapy. In the second version, mitochondria of oocytes or zygotes at risk could be subjected to corrective genome editing or germline editing or heritable genome editing, as some of the recent commissions chose to refer to this practice. This brief presentation will review the present state of germline remediation of mitochondrial DNA diseases and reflect, as time permits, on the future thereof. Beginning with mitochondrial replacement. Recent efforts to advance the cause of mitochondrial replacement therapy at the germline level focus on the replacement of mitochondria of oocytes or zygotes at risk with donated, unaffected counterparts. The leading edge of this biotechnological front, so-called pronuclear transfer, or PNT for short, and maternal spindle transfer, or MST for short, have been the subject of extensive preclinical interrogation for the better part of the last decade. The more recently added polar body transfer, or PBT for short, has yet to be explored in the same degree of detail. Mitochondrial genome editing is the new kid on the block. The prospect of precision editing of mutant mitochondrial alleles has just been reported a month or two ago using a novel CRISPR-free mitochondrial DNA base editor. The latter, which is the first of its kind, is a cytosine base editor which catalyzes the conversion of CG pairs to TA pairs without effectuating a double-strand DNA break, which is remarkable because the mitochondria has a very poor DNA repair mechanism, and so avoiding a double-strand DNA break is a great advantage in this context. Off-target activity within the mitochondrial genome of cultured cells was deemed to be low, and the same is true for the nuclear genome. The paper in question was published in Nature approximately 2 months ago. The first author, if you wish to look it up, was Mok, M-O-K, and of course associates. The senior author was, among the senior authors, was David Liu from the Broad Institute in Cambridge, who is a leader in base editing and broke the record yet again by introducing the first ever mitochondrial gene editor. Despite the progress that has been made, we are still faced with what I would characterize as residual challenges. The first, which I'm sure you all know about, is the so-called carryover challenge. This is common to all of the forms of MRT, and the risk here is that you transfer mutant, in this case, maternal mitochondrial DNA, into the reconstructed oocyte or zygote. Even the report of the first human infant born in the wake of MST affirmed the reality of the carryover phenomenon, as well as its postnatal persistence. What is still poorly understood, though, is the possibility that carried over mutant mitochondrial DNA might in fact outgrow its intact donor counterpart, and in so doing, reestablish the disease phenotype. The second challenge that is related to the carryover challenge is referred to usually as the reversion challenge. This was first noted in the oocytes of female progeny of MST-derived non-human primates. And the prospect of reversion has since been repeatedly reaffirmed. More recent studies of adult MST-derived rhesus macaques and their offspring noted a range of maternal heteroplasmy levels up to 16.6% in several internal organs. These and related observations leave little doubt as to the existence of the reversion phenomenon. What is unknown, or remains unknown, is its prevalence, its theoretical ceiling, and its informed prediction. The genome editing option, promising as it is, still must also be viewed in the context of its multiple challenges. Realizing the full potential of base editing at the germline level will still require the redress of significant residual shortfalls. Topping this list is the reliance of base editors on mitochondrial DNA replication. That requirement alone caps the editing efficiency of base editors at 50%. Moreover, mitochondrial DNA replication, which is really the domain of blastocyst stage embryos, is unlikely to materialize during earlier stages of human development, during which germline genome editing is applied. Post-interventional reversion to the pathogenic mutation must also be considered, although of course we have no information about that at this time. At this point in our scientific evolution, we find ourselves torn between uncertainty and promise. And the germline remediation of mitochondrial DNA disease, promising as it is still in a field of flux. Concerns over the carryover of mutant mitochondrial alleles and their postnatal reversion continue to haunt the field. Any and all progress in this arena will all but assure, though, that the relief of the attendant human misery, which is for the first time in sight, hopefully will materialize before too long. So that's where we are today. Quite a bit of promise, but still quite a few questions, and certainly no certainty that what we are projecting will resolve the problem or address it in a meaningful way. That still is somewhat up in the air, and studies that are currently ongoing in Newcastle in the UK, the results of which are unknown to us at this time, doubtlessly will shed a great deal of light on the practicality and efficacy, in this case of PNT, but by extension, probably the efficacy of other technologies that are in store for the correction of mitochondrial DNA diseases. So with that, I will close. And of course, I'm sure questions will arise and we can try to tackle those as we go. And hopefully that will provide further clarity. So thank you, Serena, for the opportunity, and back to you.
Thank you, Dr. Adashi. I am hoping that your talk will be summarized in your next publication, because that was quite complex and a wonderful overview of the current technologies that hold some promise. And I have to admit, I certainly did not completely follow all of it. So thank you so much for that. I do have a question right here. We're not going to answer all the questions, but I want to put up one question, and later I'll hang around, and because there's going to be there's already a ton of questions here. But from one of our embryologists, they are asking, I think, a question that many of us may have. What about using PGT and just using PGT to find the diseased mitochondria? Like, are they always in all the eggs, or can we just get the good eggs out?
So PGT has been certainly explored in this context. And if there is a challenge there, it is agreeing on the level of disease, or the cutoff level, which would define or distinguish a quote unquote normal oocyte from an abnormal one, or a normal embryo in this case, versus an abnormal one. That said, if you actually read the HFEA instructions in the UK, they make a very strong case to proceed any mitochondrial replacement procedure with PGD if feasible and if helpful. It's a little hard to tell where that situation stands. I reached out, in fact, recently to the folks in Newcastle and to the folks in Paris, to Julia Stefan, who has done a lot of work on that, and to the folks in Australia who are doing a lot of that. And my sense was that while it's being practiced, there is no uniform conviction as to the utility of PGD as a solution, if you will, to the problem. It's a selection technology, of course. It's not a therapeutic technology, by the time you get to that stage, therapy is probably too late. In other words, would not be feasible. So I would conclude by saying that we need to keep PGD in mind. PGD was and remains, I think, a part of the picture. The HFEA certainly has that on its website. But as a practical matter, my sense from the embryologists I've spoken to, or the reproductive endocrinologists I've spoken to, is that it's a little muddy in terms of practical utility, and whether or not we want to put patients through it is, I guess, a matter of discussion.
Thank you. I think Jacques now.
Yeah. Well, thank you, both of you, and thank you, Professor Adashi. And I think we'll come back to PGT maybe a little later, but the issue of heteroplasmy and determining each embryo is, of course, challenging. And one wonders about the accuracy in those cases. So certainly that's a possibility to do PGT, and it has been done, particularly in the Netherlands, in Maastricht. There have been almost as many as 100 cases done already, with quite a few babies. And so it is possible, but there is this issue of transferring embryos that have 10, 20%. I think their cutoff was 18%. Mutated mitochondria in their biopsy. And so you then wonder, well, can you get a genetic drift later on? So let's get back to that later. It's my pleasure to start the first of 3 lectures on cytoplasmic donation for infertility, which is an underappreciated topic that comes up in meetings, reproductive medical meetings, but not necessarily meetings in the mitochondrial community. And so hopefully next year, and hopefully with Professor Adashi, we will come back next year and have a session on treating mitochondrial disease in the way that he discussed. But for the next 3 lectures, we're looking at cytoplasmic donation, mitochondrial replacement therapy used to be called. There are different ways of describing it for infertility. Anyhow, that's what we're trying to do. And the first speaker is Dr. Serena Chen, and she is going to talk to us about cytoplasmic transfer. There are 2 big questions in cytoplasmic transfer. There are a lot of questions, but one of them is, of course, why? And the second one is, are the kids okay? And she's going to talk mainly, I think, about are the kids okay? So Serena, my pleasure, please.
Thank you so much, Jacques. It's a real honor and a privilege to present this work, and having worked with these patients directly, I actually have a very positive perspective on this technology, and I think this is a great opportunity to clear up a lot of the misconceptions that have arisen about this study in the last 20 years and continue today to be associated with this work. The process of cytoplasmic donation was copied by many around the globe and used clinically with success. But I think Jacques' IRB approved study at IRMS at St. Barnabas in New Jersey was based on animal data. And I think he was the first. I'm not totally sure, but I think he was the first. And it was only a handful of, it was one of only a handful of studies that really was IRB approved and had some published outcomes. Ironically, I feel that it was Dr. Cohen's transparency that actually led or seemed to lead to some of the drama that ultimately had something to do, I think, with the cessation of this research in the United States by the regulatory bodies, the FDA specifically. So I believe that this was kind of an overreaction, a negative overreaction that ultimately harmed patients and created barriers to care in an area where infertility patients already, as we know, at least in the United States, face tremendous barriers to care. We are in a country, theoretically one of the richest nations in the world, that actually 85% of people in the United States do not have any insurance coverage for this technology at all, even though millions are affected by infertility and many more need this technology in order to have a family. I think that is something that personally I would like to change, and I hope those of you here today will consider the implications of how we continue the dialogue going forward. I think we do have to be concerned about risks, but I think we have to balance that with the realization that obstructions to this research can also have negative consequences. So many of you probably know Dr. Cohen very well, but for those of you who don't, his accomplishments in reproductive science are groundbreaking. And I want to talk about those a little bit because I'm one of the authors on the paper, but he obviously, this is all based upon his work. And he's had many groundbreaking innovations in IVF and has been a huge positive influence on how IVF is practiced today, really revolutionizing this technology and bringing it to the stage today where it is hugely successful. And some of those innovations include blast cryopreservation, assisted fertilization, assisted hatching, pre-implantation genetic testing, and of course, cytoplasmic donation, which we're discussing today. The details of Jacques' very distinguished career are listed on the IVFmeeting.com website. And personally, I've known Jacques since 1999 when I came to IRMS at St. Barnabas in New Jersey as a young physician, just 3 years out of my fellowship. And I've been honored for over 20 years now to have Dr. Cohen as a mentor, a collaborator, a colleague, and a friend. So let me go ahead and present the study now. Cytoplasmic transplantation in the 1990s. Are the kids all right? Here are my affiliations and conflicts of interest. The most important part of the slide, and honestly for me, the presentation is notice my Twitter and Instagram handle. I'm basically here to get more Instagram and Twitter followers, but don't tell Jacques that. Anyway, Dr. Serena H, don't forget the H, Chen. And that is because there's so much more to talk about than the 10 minutes I have today. And I would love for you to connect with me on Twitter and Instagram to discuss more about germline genetic modification, the implications, as well as better access to care. So I hope you guys will all connect with me. So I'm going to talk about cytoplasmic donation. We'll do some background and the follow-up and hopefully next steps. The history is that Jacques, I think, came to St. Barnabas around 94, 95. St. Barnabas devoted a tremendous amount of resources to not only clinical care, but research at the time. And so Jacques and his lab team and the clinical team had the opportunity to really do some very important research. At that time, implantation rates were much lower than they are today, and multiple embryo transfer was the norm, even in donor egg and in high prognosis patients, less than 35 years old. We were doing a minimum of 2 embryos. The only time we ever did single embryo transfer, unlike today, where we do mostly single embryo transfers, the only time we were doing single embryo transfer in those days was when we only had 1 embryo to transfer. In fact, I think we were in like a National Geographic magazine cover special article with like New Jersey being the #1 place in the country with triplet births, which actually happened to be from IUI, not IVF. But we were doing multiple embryo transfer because that's really what worked. We had some inkling about embryonic aneuploidy and the association with age. Santiago Munet was there at this time and was starting his studies on fish and aneuploidy and things like that. And so we had some idea like that age-related infertility was related to aneuploidy, but we continue to see the small group of patients coming to us from all over the country and sometimes outside the country that had recurrent IVF failure that seemed to be of a cytoplasmic origin. These were young women. They did not necessarily have diminished ovarian reserve, but their very early embryonic development, despite numerous interventions at the clinical level, the stimulation level, the media level, and other interventions, all these tricks that Jacques and his colleagues had up their sleeves, they still continue to show terrible early embryonic development. And so we saw these people from all over the country. The first step we take with failed cycles back in those days was, well, the St. Barnabas lab was one of the best in the country, best in the world. And we were like, well, once these embryos get into the IRMS lab, they're going to look better. They're going to look fine, and we're going to get them pregnant. But there was still a group that were resistant to all our interventions. And this group really puzzled Jacques. And he saw an analogy with this mouse model. So it's called the mouse development block model. And those mice made embryos that just like arrested at a very early stage of development. And it was a cytoplasmic factor. And that was the basis and the inspiration for this particular study. Jacques postulated there was some magic juju in the cytoplasm that was not identified in the mouse and not identified in humans, but we knew something was going on with these patients, and it was something not related to age and not related to the culture environment or other factors. So that's where we started doing the cytoplasmic transfer by injection. So we had a target group, repeated IVF failure. We specifically excluded people over 40 because we did not want advanced maternal age and nuclear aneuploidy to be a confounding factor. And we are trying to overcome poor development. So these are these embryos that, before day 4, before compaction, just started to fall apart very early in the dish. Tremendous amounts of fragmentation, slow division. We delivered the donor cytoplasm through the ICSI needle at the same time that we delivered the sperm. And this had to be a synchronous donation. So we used fresh donor egg cytoplasm and a fresh recipient egg. And we had to synchronize these cycles. So all these patients had at least one, maybe all of them had 2 IVF failures at St. Barnabas. So you do one routine St. Barnabas IVF cycle, your embryos look terrible. Then we do all the interventions, do another cycle, it still looks terrible. Then we offer you the cytoplasmic donation protocol. Many, many people declined because it was it's experimental and they did not feel comfortable. But the people that agreed had to then go through the donor screening program. They selected a donor, and the donor eggs, the excess eggs, were then used to create donor embryos as a backup plan. So then we started the donor and the recipient kind of at the same time. If they had cycled previously, we would kind of know about when the trigger point would be. So we tried to coordinate so that we could maximize, we could coordinate the HCG time, the HCG day of the donor and the recipient at the same time, which sometimes was quite tricky. So it was clinically challenging to set these cycles up. Then on the day of the retrieval, we took the donor eggs, and we usually the cytoplasm from the donor eggs, we could get 1 donor egg to donate to 2 recipient eggs. And usually the donors made more eggs than the recipients, even though the recipients were not diminished ovarian reserve patients, but they did tend to be older, and they did tend to have less eggs than the donors. So we usually had extra donor eggs left over, and then the partners of the recipients' sperm was injected into the excess donor eggs, and those embryos were then watched, cultured, and frozen as a backup. So let's see, that's this slide. So that's how we did the process. This next slide is slow. I should have started. It's a big, okay. So the top 2 frames are the donor egg. The bottom 2 frames are the recipient egg. And you can see we just use an ICSI needle. This is the holding pipette. We go into the donor egg and we pull out some cytoplasm. And let's see right here, this arrow just shows like we had a mark on the needle in order to know about how much cytoplasm to pull up. We were aiming for about like, I think 10 to 15% of the volume of the cytoplasm. I'm not sure how Jacques came up with those calculations. He can pot for you cytoplasmic donation geeks. We could probably, talk amongst ourselves later. Then this is the recipient egg and in the holding pipette. So just so you know, the sperm is actually in the pipe, in the microneedle before we pierce the oolemma of the donor egg. So the sperm is in there. We pierce the oolemma of the donor egg and then pull back with suction to a certain point so that, the cytoplasm is here and the sperm is back here. And then here is, we've just injected the recipient egg with the microneedle, and there's the arrow is pointing to the little sperm that's been injected. And here is like a little photoshopping just to highlight the area of cytoplasm that we left behind into the recipient oocyte. So this was a prospective pilot study. There was no randomization, and the average number of previous failed IVF cycles is about 4.8. So this was a group that had failed, as I said before, a lot of IVF cycles. All of them failed at least one or two IVF cycles specifically at the Barnabas lab, not everybody, and then possibly some other failed cycles at other labs outside. There were 37 attempts in 33 patients and 12 deliveries, 17 babies. And this was reported in previous publications, 2 implantations with XO, 1 miscarried, and 1 was selective reduction of twins, with the remaining twin doing well and being delivered. 1 male twin at 18 months was reported by his parents to have borderline PDD NOS, pervasive developmental disorder, not otherwise specified. Later, a false positive because he's now doing well with, does not really have any evidence for PDD at the time, and really at 18 months, a little bit early to be diagnosing PDD. This part was very interesting that Jason Barrett followed 8 mothers around and collected samples from them and found from the babies, found donor mitochondria, persistence of donor mitochondria in 2 out of the 8 babies. There was also a report that I think it was either Brenner or Barrett, and Carol Brenner, I see you're here. Hello. The 1 out of the 6 pregnancies that we were able to collect amniotic fluid on were tested for donor mitochondria, and 1 of the 6 was found to have donor mitochondria, and that baby later, when they delivered, did not show persistence of the donor mitochondria. So that was kind of interesting. It is important to remember, Jacques always reminds me that the technology we had at the time to test for mitochondrial heteroplasmy was much more primitive than it is today. And if we were to do this study today, we might find something different, maybe a higher rate of persistence of donor mitochondria, possibly because these assays were relatively non-sensitive. So we're looking for DNA fingerprinting on the donor mitochondria. Remember, these are not people affected with mitochondrial disease. These people were all, other than their infertility, were basically healthy. And then let me talk with you about our follow-up survey study. So this was published in 2016 in RBM, and it was a preliminary survey study. We had basically in 2001, when Jason Barrett published the mitochondrial heteroplasmy persistence, there was a huge outcry at the time, and very, very controversial. And it was all over the scientific and lay press. and that created a big outcry. I remember very well. I was just a busy young doctor at in New Jersey, and Jacques study was talked about on the CBS. I remember the Jacques study was talked about on the CBS Evening News. I think Dr. Arnie Cohen, not related, uh, from University of Pennsylvania, said that this was an unethical study, despite the fact that it was IRB reviewed and there was follow up, and our phones just were blowing up. Everybody was calling from all over the country. And the most common phone call that I got was, Dr. Chen, I saw you guys on the evening news about that new study, that unethical study. Can can I be in the unethical study? Because that's what I want. I want a baby and I want to be in the unethical study. So this was the response from the patients. It was certainly very different than the scientific community. And I think that deserves some consideration. Hopefully we'll talk about that later, or you can follow me on Instagram or Twitter to talk about that. So anyway, follow-up study. Let's go through the data. So we so many years later, we basically, after 2001, the FDA said, you guys have to fill out an IND. We're concerned about this study. St. Barnabas lost funding. They cut back all the FTEs. We were not able to do any more research. And of course, there's no federal research available. So the research essentially stopped. And so many years later, Jacques said multiple times, Serena, we should really follow up. We should really follow up. And finally, we decided we would go before the IRB and try to do follow up. And the IRB had numerous concerns about the follow up, about invasion of privacy, about coercion, all kinds of concerns. And they wanted us to go very slowly and gently. And so we started with a survey study, just a questionnaire. And there was a lot of resistance and a lack of response. And it took us like almost 2 years to get all of these parents to respond to us. And indeed, one of the parents of the quads, did not respond to us. So we had 12 out of the 13 couples or patients participated in the baby follow-up study, and we asked them by questionnaire detailed questions about disclosure, pregnancy, birth, health, academic performance, and we found that all of them had very positive experiences, despite the fact that they were not so responsive in the beginning. It turns out, I think Marianne can maybe clarify this for me, but it's not that they were necessarily hiding from us. The quads were hiding from us, but everybody else was not necessarily hiding from us or trying to avoid us. They were just super busy. So these kids at the time of the survey were 15 to 20 years old. The parents reported prenatal development, delivery uneventful, school grades good to excellent. 13 out of the 14 children had normal BMIs. We have 14 kids in this group because the 14th kid was one of a set of twins where we did a mixed transfer, one cytoplasmic donation baby and one straight egg donation baby. And we think we know which one is the egg donation baby, or the mom thinks she does, but they have not actually been DNA fingerprinted, so we don't know for sure. So we did include that twin, the straight donation twin, in the outcome data. No current developmental disorders. One child has ADD supposedly, but is getting straight A's. So, and only one out of 12 that has actually disclosed the use of cytoplasmic donation in their child's conception to their child. So this I found to be one of the most compelling things that really surprised us about this study. We kind of knew the kids were doing well because a lot of these families actually stayed in touch with Maria Jackson and Claudia Pascal, two of the authors on this paper, our donor egg nurse and our reproductive psychologist, and so they knew that these kids were really thriving. or at least some of these kids were really thriving. So the good outcomes and the good health of these children was really not surprising to us, but this disclosure rate really was. And there's not that much literature about disclosure rates in reproductive medicine. This Taladini et al. In Human Reproduction in 2016 did a nice job of doing a big review and a meta-analysis. And so that's why these ranges are really wide. I've extrapolated this from one of their tables because they go through disclosure at a young age, disclosure at a later age. There are differences. So they have a lot more analysis. Definitely disclose, planning to disclose. There's many different things to talk about, but I simplified this to, and this is what I came up with, that in general, today at least, with more acceptance of IVF worldwide as a routine way to conceive, parents are typically in the 90 to 100% range for disclosing to their children that they were conceived through IVF. After egg donation, it's lower. After sperm donation, the trend tends to be even lower, as low as 25% in some of these studies. But after, in our study, we had an 8.33% disclosure rate, obviously a very small N, but this was remarkably very low. And we have a lot of theories about why there are differences. One thing is we have a big selection bias. This group of patients specifically were trying to avoid egg donation. They had all been told they needed egg donation, and they were specifically entered the study to try to avoid egg donation. And we also think that the uproar that occurred, the controversies that occurred in 2001 in the lay press probably discouraged these parents from disclosing this technology, the use of this technology to anyone. Interestingly, the other interesting thing is, I think, especially for the embryologists, is that these were horrible looking embryos before cytoplasmic donation. After cytoplasmic donation, morphology was still horrific. And that's probably why we ended up with quads. The quads specifically, we were, I've looked at those pictures. Embryo morphology was horrible. And I think at the time we did the transfer, nobody thought there was going to be any implantation, and yet all 4 of these very poor quality embryos implanted. So that I thought was another thing that was quite interesting. So obviously the N is very small, but when you look at our data from our program, I think this is in the 1990s, we have like 8,500 embryos, 2,500 transfers. In these first 5 columns, where we're looking at the impact of previous failed cycles upon the pregnancy rate and upon the implantation rate. And in your first cycle of IVF, that's your best chance of succeeding. Very high pregnancy rates, very high implantation rates. Remember, we did multiple embryo transfers here. After you failed one IVF cycle, at least at St. Barnabas, the pregnancy rate dropped dramatically and continued to drop the more cycles you failed. And yet in this group of cytoplasmic transfer here, where all these patients had like 3 to 10 previous failed cycles, an average of 4.8, we had a much higher than expected pregnancy rate. So hopefully I didn't take too long. I wanted to thank everybody that participated. It obviously took a big team to do this, and thank you for having me.
Thank you very much, Serena, and thank you also for the kind words in the beginning. That did make it many, many minutes longer. So, but nevertheless, thank you very, very much. There are a bunch of questions here, and one that I think is of particular interest to just your last few slides is why is it so difficult, you think, for some patients to accept just egg donation as a good alternative when nothing else works? Why are there some patients that are refusing this? Because I've never met an egg donation patient who later comes back and say, well, I'm sorry I did this.
So it's the reproductive psychologists tell us that not only is infertility extremely traumatic, emotionally traumatic, but it is also moving from your own eggs to egg donation is experienced as a profound loss, similar to death of a loved one. And until the patient truly processes that loss, which can take many years and sometimes never happens, they cannot really fully accept egg donation. And our society is conditioned to think of nuclear genetics as the essence of our being. And of course, now that we're moving forward and we're learning more about epigenetics, maybe that will change. But we think, that is the most important part of the cell of our being is the nuclear genetics. There's so much emphasis that culture and society places upon that. And I think that's why it's so difficult to let go.
Yeah, it's fascinating. Thank you again. And we'll move on to the next speaker. Thanks, Serena. It's now my pleasure to introduce our third speaker for today, my friend Pavlo Mazur from the Nadia Clinic. Pavlo earned his bachelor degree in virology in 2009. And so you may remember him from when we started the ICE3 series, because we needed virologists. And so he came forward, and here he was, a virologist and an embryologist. It's a unique combination of experience during those early pandemic days. And now about 8 months later, we're not in the clear yet, obviously, in deep trouble. But we'll get out of this because there's considerable hope, and we're in this mid-pandemic state. And so I had the pleasure of writing a paper with Pavlo over the last, 6, 7, 8, 9 months. And so he was my private virologist for all this time, and he gave me weekly information sessions of what he thought about all of it. So he's a virologist, but that is not enough for Pavlo. He also has a master's degree in genetics, and that is in 2011 from Taras Shevchenko National University of Kiev in the Ukraine. And he started immediately after that, he started doing clinical embryology. So very quickly, in a miniature fashion, did what Ailey did in her whole career. And he moved. And of course, virology and genetics has been extremely helpful, I'm sure. So he looked particularly at time-lapse incubator aspects. He joined Nadia Clinic as a trainee embryologist in 2011. And since then has studied time-lapse further. He has had an interest in AI for embryo selection. And in late 2015, he developed, and this was after about, I think, 14 months of intensive preclinical experimentation, because you really have to get very, very good at this. Earlier on talked about carryover when you have patients of mitochondrial disease. And so what the aim is even when you do it for infertility, when you do cytoplasmic donation, and Pavlo looked at the pronuclear transplantation aspect, you try to diminish or minimize the carryover and keep the mitochondria low. Also for just the research purposes, because we feel that in infertility that plays less of a role, because there shouldn't be any mutated mitochondria, not mutated to the extent that this would cause mitochondrial disease. So the first embryo transfer after the reconstitution of pronuclear embryos was successfully performed in April 2016, and a healthy baby was born on the 5th of January 2017. I think that was the second baby born from mitochondrial replacement therapy, or in this case, cytoplasmic donation. So Pavlo, looking forward to your lecture.
So thank you so much for such introduction. I made a rehearsal on this presentation, and it took me like an hour to do so. So I'll try to speed up. Okay, I declare no conflict of interest. Here in Ukraine, we have some permissions to do such procedures, and you should be aware that in Ukraine, it's not like in other European, for example, countries. But after all, we have this legal opinion which says that, well, for now, And at the time we started, we didn't need any additional agreements by public authorities, but still we have this work done for Ministry of Health. We have the conclusion of bioethics committee of our National Academy of Sciences, and we have the permission of IRB panel by our Ukrainian Association of Reproductive Medicine. So we have some, and we'll move on. When we started with patients of advanced maternal age, but very fast we've moved to patients of much younger patients, and I'll show you why. In this presentation, there are two very important tables and one not so important. But as for indications, prevention of mitochondrial diseases, we didn't have here in Ukraine, so it was questionable. And it's still questionable. And again, I will show you later why. So we've chosen patients with complete embryo arrest. We'll talk a bit about this and about repeated implantation failure, even in young patients, even when you transfer the euploid embryos. So embryo arrest, I think that embryologists know well this condition. When you have day 5 or day 6 or even day 7 of patients' embryos and they do not develop to the blastocyst stage, you can call it embryo arrest. And if you have such condition in recurrent cycles, well, that means that you have this condition. Why? Yeah, we've made some our own retrospective investigations of how many patients do we actually have with this. And as you can see, for 12 years, the number is around 3% totally. But when it's among the young patients only, when you add the age to this equation, you will have much higher percentages of such cases. And if you look at the younger or normal responders among these younger patients, you will see that around 2% of patients that have this maturation of or total embryo arrest condition. So these are our patients for nuclear transplantations. There are a bunch of known genes that can lead to such condition as total embryo arrest or maturation arrest. I'll just briefly show them. If you watch this presentation online, you can stop at any time and watch them closely. Yeah. So this is a work where we participated, our laboratory, and we found among our patients at least 2 patients with TLES6 mutation and another patient with TUBB8 mutation, which caused the total embryo arrest. And it's my pleasure to say that the patient with TUBB8 mutation, she conceived and she delivered a healthy baby, and I'll show you it later. So as interconclusions, we can say that, well, embryologists know that this condition exists. We know that some patients, at least in Ukraine, in our clinic, we have at least 2% of such patients have this condition. And unfortunately, we cannot say about all the reasons about it, but at least some of them. And if you have the genetic lab in your clinic or somewhere nearby, you can have a panel of those genes. And when you find one, you can say that, well, you can propose such procedures as nuclear transplantations to those patients. So in some case, we really might help them. In general, we can divide all the nuclear transplantation methods in two main groups. It's before fertilization, as you can see here, and after fertilization. And again, in our clinic, in our lab, we perform 6 out of 7, except for prenuclear transplantation, nuclear transplantations here. But the topic here is prenuclear transplantation, so we will focus on it. It's just set up as everything, and of course the working station, everything you need to perform the PNT for your patients. It's not so much, but well, I think that each lab can afford this setup. A brief scheme here, and I will show a video later of this, how it's performed. So first of all, we need to enucleate and then transfer the karyotype or karyoplast inside the enucleated zygotic cytoplast of a donor. Here you can see the reverse reconstitution it's our control group where we put the donor genetic material. It's donor pronucleus and partners, pronucleus partners of a patient. Pronucleus together inside the patient's cytoplasm. We needed to, well, first of all, we don't want to kill any excess embryos or destroy the potential zygotes. So we want to make the chances equal for both cytoplasts. That's one of the reasons why we're doing this. And that's a good control to say to us what went wrong. Either it was a karyoplast or genetic material of patient or cytoplasm itself. And it worked well, and I'll show it in those one of the important table. As for me, I like to say that this scheme reminds me the cut and paste process in a Word editor. You just need to cut the genetic material from a patient cell and to paste it or put it inside the donor cytoplasm. So all the schemes for all the nuclear transfers are almost the same. Here are PNT-specific potential advantages and disadvantages. So here you will work only with fertilized eggs, so the number of eggs much lower than you're making it with M2 oocytes or GV oocytes or M1 oocytes, and you have the picture at the step forward. Then you working with unfertilized oocytes because you don't know if they will be fertilized. No need to use special polarizing microscopy. It could be very expensive for some labs, but for Olympus it's not so expensive, it's quite cheap. And you can use for PNT 1 PN or 3 PN as well. And we have 2 kids born after using either 1 PN or 3 PN. So it's a potential advantage, and I can say that it works. That's why I put it here. And usually when you can trace the way of the female pronucleus from the second polar body, you can say that the origin of PN is well known. As for potential disadvantages, as far as we're performing this procedure at 12 hours after fertilization, you should adjust your schedule to perform this procedure either during the night or at the very beginning of the day. So it's not very convenient because you need to have the pre-warmed, prepared equipment, dishes, and all this stuff to perform the procedure, and the procedure itself takes time. So it's really inconvenient to do so. Another thing, we don't know if we are interfering with all these processes that occur in pronuclei, which are DNA replication and simultaneously demethylation of DNA, telomere elongation. So far, we know that our kids are healthy, they're good, and So it's only potential disadvantage, but we need to follow them up to, I don't know, maybe 18 years, 30 years, 50 years, who knows. About centrosome issue, it's an issue only PNT specific because you accidentally can get rid of centrosome and you will have the abnormal cleavage or absence of cleavage of your reconstituted zygote. So you should be very careful while performing this manipulation and not to suck all the cytoplasm, but to leave a small amount at least to be sure that you haven't get rid of the centrosome. It's highly good thing to remove the polar body prior to, or the second polar body as well, prior to leaving the reconstituted zygotes inside an incubator, because they can be sucked back to the reconstituted zygote and create an additional pronucleus or pronuclei. And you don't want to do this. And of course, for those who believes that my manipulations before meiosis can change something or repair something, or you can somehow interfere in a good way with the genetic material of those cells. Of course, it's not so good news that when you perform in PNT, it occurs after meiosis, so you cannot repair anything at that step. Here is a brief video. It's well-known one, because it's not because I don't have another videos. I have a bunch of them, but this one is a more clear video. So you can see the removal of 2 PNs from a patient zygote. And reconstitution itself. So you just need to put this pronuclei inside the enucleated zygotic cytoplasm of donor. That's all, quite easy. So that's the first important table here, and you will be able to pause this video and look closer on it. But the main conclusion from this table is that PNT results are very age dependent. So again, it's not suitable for patients of advanced maternal age. I'll try to make a pointer here. Nope. Okay, never mind. Anyway, as you can see in a group of young patients, we have 9 kids, and in a group of advanced maternal aged patients, we accidentally have 1 kid. So, but for reverse PNT, as you can see here, the development rate is quite high, and for younger patients is quite low. It means only that we've included the right patients to this procedure, because they really had some cytoplasmic problems. Which one, we don't know, but still they had some cytoplasmic issues, and we helped them with nuclear transplantation. For advanced maternal age or intermediate age here, patients, you can say that, well, the problem of their underdevelopment, or I mean the embryos not developed to the blastocyst stage, was not quite in their cytoplasm, because most of those blastocysts from reverse PNT group, they were euploid. So the number of euploidity is around 70% or so. And you can say that the cytoplasm is good, even if you have these advanced maternal age patients. The problem was within their karyoplasts. And for younger patients, as you can see, everything worked perfectly. Their embryos were euploid, and we have a lot of pregnancies. So this investigation was performed for 2 years, from 2015 until 2019, and totally we had a group of 29 patients. That's not so important table. It shows only that how many zygotes survived of our manipulations. And as you can see, it's not age dependent because the percentages are the same among all the groups. So our precious, this interesting list, because you can see here the two snowflakes. They mean that we've used frozen donor oocytes to perform the procedure because we didn't have a fresh donor oocytes, and still it worked. And one girl here with 2BB8, she is no longer with this gene, so we get rid of this mutated gene from the population, which is quite impressive for me. So her mother was a carrier of a mutated gene, she was infertile, and then we've removed this gene from the population because her daughter is completely healthy. And for our surprise, the next boy, he has a sibling which was born this year. And we can say that we've done our work very good because we've treated the infertility. And this patient, she was infertile for many years, and after delivering a boy, after PNT, she conceived naturally. So she is healthy now, which is good news. And the second most important table and climax of this presentation, actually, you can see here the cases. They were numbered from number 1 until to number 29, and the kids were born for these cases. So for A and for B, it's actually here kids number 1 and number 10, which were born by the one woman. So she gave birth to the girl, the first one in the world you can see on right. And then she came to us for a second child, conceived and delivered a second baby in September 2019. So she is some sort of our hero. And here you can see those babies, they are 4A, it's a girl, and 4B. And you can see the heteroplasmy level, which is quite high for the boy. But I need to stop here and say the next thing. The setup was the same. The zygotes were done in the same time, on the same working station, by the same person, with the same donor cytoplasts. So everything was the same for these two cases. And in a girl, we have this very low heteroplasmy level. It means that maternal DNA, mitochondrial DNA, it's on the traces of these mitochondria. But in the boy, you can see that almost all of the mitochondria are maternal. Probably in several years, there will be only traces or no traces of the PNT was done in this case. So this boy will be completely out of donor mitochondria. So he will possess only maternal mitochondrial DNA. And it means that distribution of mitochondria is random. And even if you have very small, tiny karyoplastic drop with a very low loadout of mitochondria, it means nothing because the distribution can be random. And it's okay, it's fine when you say about healthy mitochondria, but when you have affected mitochondria, it means a lot. It means that it might be dangerous to perform nuclear transfer in case of mitochondrial diseases, because you never know. Even if you have the load of maternal mitochondria at less than 5%. After several years, you still can have a very high level of maternal mitochondria, affected maternal mitochondria. And here I want to put a period, full stop, about the mitochondria genetic incompatibility, because the 85 proteins in respiratory chains, and only 13 of them are coded by mitochondrial DNA, and all the others are coded in nuclear DNA. So we know that more than 1,000 genes are responsible for living of the mitochondria, for their fusion, for their fission, and for all the processes, even for mitochondrial DNA replications. All these genes are in the nuclear DNA. And so far we inherit the half of our nuclear DNA from our fathers, and they have nothing to do with the mitochondrial DNA of our cytoplasm. There should be a mechanism for adjusting the mitochondrial genome to the nuclear genome, which means that when the embryonic genome activation occurs, a lot of mitochondria die because of this adjusting, because only the fittest, only the suitable, only the mitochondria which can have good relations with nuclear DNA will survive. So from my perspective, there should be no such thing as mitochondrial and nuclear incompatibility, because, well, nature makes this for millions of years and we're still here. So conclusions. We know that strict indications are needed, and the nuclear transplantation should be the last stand for such patients. There should be no other alternatives except for the anti-procedures to conceive. And only in this reason we can propose such patients to go through this procedure. As we can see here, the mitochondrial diseases might not be a good indication for PNT, as well as all the other nuclear transfers or transplantations. We believe that we have this technology to remove the maternal mitochondria completely, but we need to go through more tests, and then we will be able to show you this and to share with our results in this field. And that's true that the age is very crucial. We cannot repair what's already damaged, even at GV stage. As I said, we perform all types of nuclear transplantations, and we've seen the same pictures for any type of nuclear transplantations. Even when you transfer the GV germinal vesicle, you have the same results: high level of aneuploidies for advanced maternal age, and low level of aneuploidies for younger patients. And I believe there will be more children after PNT. And here is, on this slide, is my favorite joke. It's never get old. When journalists say about the three-parent baby, not person baby, but three parents baby or babies. So here are your three parents. It's Dr. Viktor Vatsilovsky, Dr. Valery Zukin, and some random guy in white sweater. So yeah, thank you for your attention.
All right, thank you, Pavlo, for this terrific presentation. Of course, it's been a tremendous amount of work in those last 5 years, and it's amazing you pulled this off. Compliments to you and your team. Got questions here, and just one quick one because we're running a little late. So one quick question from Debbie Monjan from Marseille, from France. Any comment about the learning curve How long is this possible to learn for everybody, first of all, and how long would it take?
Actually have no idea because it's a combination of classical embryology work and well, it's a small addition to it. So the learning curve could be either, probably it's person dependent. It could be very fast or it could take forever. I think it's person dependent. As you can see, the setup is quite simple.
Yeah. I think you're being modest. It took you, I remember you told me, it took you 14 months. Of course, you had no peer. This is from scratch, I think. So, yeah. So, but it is pretty intense, and you're doing this in the middle of the night because of the timing of Pronuclear and all that. So, yeah, really well done. Over to you, Serena. Thanks again, Pavlo. Over to you, Serena.
Thanks, Jacques.
Thank
I would say this is kind of like, it's like cytoplasmic genetic surgery, essentially. And so I think probably possible, but sounds technically very difficult. So thank you again. Now it's my great pleasure to introduce our last speaker, Dr. Nuno Costa-Borges. And Nuno Costa-Borges is an embryologist who has been involved in both clinical and research practice for over 18 years. As a PhD fellow of the Portuguese National Funding Agency for Science, FCT, Nuno spent several years developing strategies to improve the efficiency of animal cloning, having accomplished the first animal successfully cloned in Spain in 2009. After he finished his PhD at the University Autonomous of Barcelona, he joined the embryology team of IVI Barcelona, where he has worked as a clinical embryologist. Later on, he co-founded Embryo Tools, which I think is one of the sponsors today, where he has been devoted to offering QC tests, training, and consulting services to the IVF community. Additionally, as scientific director of Embryo Tools, Nuno has been committed to the development of new IVF techniques, such as the flicking method for the biopsy of blastocysts, and he has made maternal spindle transfer into an art form. This is another really amazing kind of cytoplasmic microsurgical technique while doing clinical cases in Greece. Like Pavlo's approach, the intervention is aimed at preventing the transmission of mitochondrial diseases or treating infertility problems related to poor oocyte quality as alternatives to conventional egg donation. Nuno's work has led to several peer-reviewed publications, and he's been invited to speak at numerous international conferences and scientific meetings and awarded many prestigious prizes and grants, especially the ASRM, American Society for Reproductive Medicine Congressional Prize Paper in 2017 and the IVI Foundation Award in 2019. He has produced embryos that resulted in the world's first baby after maternal spindle transfer. And Nuno Costa-Borges, I'm very much looking forward to your presentation.
Thank you so much, Serena, for your kind introduction. And on behalf of Dr. Gloria Calderon and myself, we'd like to thank the opportunity for presenting our work in this session and congratulate you also for the success of this initiative. So I'm going to present the work we've been doing with maternal spindle transfer. This is a technique that, as it has been said before, has been originally proposed to prevent or to reduce the risk of transmitting mitochondrial diseases. But we thought that it could be maybe useful also for the treatment of infertility problems related to poor oocyte quality. And this is the work I'm going to present today. So it's well known, IVF treatments have evolved, and we can now circumvent many infertility or subfertility problems. However, the efficacy of these conventional treatments is still limited by the number and the quality of the oocytes that are used. And it is frequent, as Pavlo explained before, that in some cases, IVF patients, some of them still young, most of the times, present what is called massive embryo development arrest. That is mainly a problem that is related to the fact that the oocytes, after being inseminated, do not develop into good quality embryos after fertilization. So usually this problem tends to persist even if the treatment is repeated several times. And in the absence of a male factor, usually this problem is attributed to poor oocyte quality. Usually this is a very frustrating situation, not only for the patients that see their hope of having a child genetically related to them diminished, but also for embryologists or medical doctors that see how they feel that cannot offer these patients alternatives to enhance this embryo development problem, other than, for example, oocyte donation, which is sometimes difficult to accept for these patients, especially if they're still young. When we talk about oocyte quality, this is usually defined as the potential or the competence of the oocyte to develop into a good quality embryo with chances to implant and sustain a pregnancy to term. And this developmental competence is usually dictated by nuclear factors and also cytoplasmic factors that are present in the oocyte. In particular, there are lots of studies showing that the cytoplasm that contains the organelles, mRNAs, ribosomes, and calcium storages, many factors that are crucial for the cells to divide. So deficiencies at this level, like in mitochondria or other cytoplasmic factors, have been indicated as a principal cause for this poor embryo developmental competence. As Dr. Chen explained before, there were strategies trying to enhance or to enhance the cytoplasm of these poor quality oocytes in patients that have passed through several previous IVF attempts that failed. One of these strategies was the one described by Dr. Chen, proposed by the group of Dr. Jacques Cohen by the end of '98. That was mainly cytoplasmic transfer work that Dr. Chen presented before, which is showed to offer some improvement in terms of implantation and full-term developmental rates. So we can probably say that this strategy of transferring a small amount of cytoplasm from a donor into this poor quality oocyte could ameliorate the cytoplasm or mitochondrial function in these poor or quality oocytes, but probably only partially, as the defective factors that might be present in these oocytes will still remain there, and they will still be passed on to the embryo. Nowadays we have technology that allows us to replace completely this cytoplasm, as Pavlo explained, with pronuclear transfer. And because we are able to replace the complete cytoplasmic machinery, we think that this offers a greater promise for these patients of having a child genetically related to them. And this is mainly the concept of the mitochondrial replacement therapy. So there are different ways of doing it by transferring the nuclear genome of an oocyte or zygote into a healthier cytoplasm from a donor. Our group has been working with maternal spindle transfer because we believe it can offer some advantages compared to the other techniques. So this technique is based on using metaphase 2 oocytes, both from a donor and from a patient. And we start by removing the meiotic spindle, what we call oocyte enucleation of the donor's oocyte. And then once this oocyte is enucleated, we transfer the meiotic spindle from the patient's oocyte with the minimum amount of cytoplasm possible around the spindle to this previously enucleated oocyte from the donor. Once reconstructed, this oocyte can be inseminated by ICSI, and if it develops in a good quality embryo, can biopsy it, and it has the potential, this embryo, to develop into a healthy baby that would be genetically related to the patient, and only the mitochondrial DNA would belong to the donor. So this is a procedure with a long learning curve that requires good micromanipulation skills. And if it's properly optimized and performed carefully, it's associated with very low mitochondrial DNA carryover. Apart, offers the advantage, as Pavlo also explained, that all the intervention in the oocyte is performed before the oocyte is fertilized. So we are manipulating no embryos, but oocytes, which from an ethical point of view sometimes is considered a positive aspect. And also another positive thing is that because those sites at metaphase 2 stage are at metaphase 2 stage, they are arrested at this stage, we don't need to speed up or to rush. So it's easier to coordinate the spindle donor oocyte with the recipient cytoplasm. So our group has been working on this technique since 2015 to test the hypothesis that we have, whether maternal spindle transfer is indeed effective and safety technique for overcoming embryo development arrest and enhance oocyte quality. This was actually an idea from Dr. Gloria Calderon that she had in mind since many years ago, or more than 20 years ago. And it was not until 2015 that we had the opportunity to explore it. So we started with a proof of concept in the mouse model, and by testing first on optimizing first each step of the procedure with a mouse strain, an hybrid mouse strain that is known to have good embryo development in vitro. And what we did was to perform the technique in both fresh and vitrified oocytes, thinking on a potential application, clinical application of this technique. We thought it would be sometimes difficult to coordinate of having fresh oocytes from the patient and from the donor on the same day. So we wanted to explore which combination would result better. So we saw that it was possible to transfer the meiotic spindle from one oocyte to another without impairing the further development of the oocyte after reconstructing, and that the best combination was when we're transferring the spindles from vitrified or fresh oocytes into fresh cytoplasm. Then we, knowing that the technique was working well, we decided to purchase a mouse strain that is known to have a very poor embryo development in vitro and in reproductive performance in general. That is the New Zealand black strain. So in a clinical context, this strain could represent this type of patients that have a poor embryo development. And what we have seen, very interesting, was that by transferring the meiotic spindles from this very sensitive strain into the enucleated oocytes from the hybrid strain that is known to have good embryo development, we were able to increase blastocyst formation rates very, very significantly, more than 10 times when compared with the non-manipulated controls from this strain. So from 5% blastocyst formation rates up to 50% blastocyst formation rates in the spindle transfer group. Then we saw that these blastocysts were able to implant and to develop to term with very good results and produce mice. And then we also found interesting to study the levels of heteroplasmy in the embryos at different developmental stages. So we performed biopsy 2-cell stage on the second polar body, cleavage, day 3 cleavage stage, and also blastocysts, and we compared the heteroplasmy levels in the embryos and in the biopsied samples. And very interesting we found that in the polar bodies, most of the mitochondria that are co-transferred with the spindle are expelled in the second polar body. So we found the highest levels of heteroplasmy in the polar bodies when compared to the complementary embryos or the embryos and the biopsies on the different, on the other developmental stages. Then from the mice that we got from spindle transfer, we did some analysis in the most important organs, and we have seen that also the heteroplasmy levels were low, between 2 and 3%, which for the mouse species is quite good, attending the size of the meiotic spindle in this species in relation to the size of the oocyte. So we have seen that the different organs, the heteroplasmy was stable. And then we crossed the mice to check their fertility condition, and we followed these mice up to 5 generations. And again, very interesting, we found that after the second generation, from the third generation, there was no heteroplasmy detected in these mice. Apart of that, to ensure also the safety of the procedure, we were requested by the reviewers to perform histological examinations in almost all organs of the mice that we have generated. So we did these histological examinations, and all organs showed to have normal morphologies. So this is a study that we published. It was a long study, but we published finally it this year in eLife. Then we wanted to do preclinical validation in human donor oocytes before starting doing real cases. So we did this preclinical validation in Greece in collaboration with the Institute of Life in Athens, and also in collaboration with a group of Dr. Dagan Wells from Oxford. We did this analysis in human donor oocytes to fine-tune all the technical steps of the procedure. And so we did we tested different nucleation and fusion protocols to optimize the technique. And we've seen with the best protocols, we were able to enucleate and reconstruct the oocyte with rates over 90%. And then once reconstructed, these oocytes were also able to fertilize, to develop the blastocyst stage, to have normal morphokinetics, or similar to the controls, as well as euploidy rates comparable to non-manipulated oocytes that were used as a control. Also, we had the opportunity also to test again whether it was feasible to work with fresh and vitrified spindles, and we have seen that yes, it is possible, but the best results were achieved when we used fresh cytoplasms. Apart of that, we also wanted to test the carryover levels, and in 30 blastocysts that were generated in these experiments, we determined the mitochondrial carryover levels, and that were lower than 1%. So the studies in the mouse and human oocytes donated for research allowed us to confirm that the technique was working well, and provided also some reassurance data concerning safety. So we decided to go on and to perform a first pilot trial. This pilot trial, of course, was authorized by the Greek National Authority and approved by the hospital's IRB. We also obtained informed consent from both patients and the donors. And we've been delivering progress update reports to the National Committee very regularly. And because it was an experimental procedure, all the treatments to the patients were offered free of cost. So the pilot trial was registered in this British ISRCETN registry, and it was addressed to women also under 40 years old that have passed through several previous IVF attempts that failed because of problems associated with embryo development arrest attributed to poor oocyte quality. So all those cases that we doubt whether it could be a severe male factor were excluded or were not included in the pilot trial. And also we didn't want to include women over 40 years old because as it has been explained before, our hypothesis is that with the maternal spindle transfer, by repairing or replacing the cytoplasm, we may be able to enhance the embryo development, but it's not likely that we can reduce the prevalence of chromosomal abnormalities that are the main factor of infertility in patients with advanced maternal. So the pilot trial was addressed to 25 patients. And I'm going to show you here some videos on how the procedure is performed. This is, oops. Not sure why this is not working. Now. Okay, so this is the enucleation procedure. This is performed in a microscope with polarized light that allows us to locate the meiotic spindle and then to remove it with as little as possible cytoplasm surrounding the meiotic spindle. This is another video of the reconstruction step. So basically the karyoplast is transferred very gentle to the perivitellin space, and after a few minutes it will fuse with the cytoplasm. So we have recruited 25 patients with an average age of 37 years old that have passed through a total of 159 cycles without any pregnancy being established, and with an average number of previous IVF failed attempts of 5.6. We used a total of 123 metaphase 2 oocytes. In average, 4.4 oocytes were used for spindle transfer per patient. So from these 123 oocytes, 113, 92%, were successfully enucleated and reconstructed. From these, 76% were correctly fertilized, so showing 2 pronuclei and a second polar body. And from these, 60% developed into good quality blastocysts. So we biopsied these blastocysts, and we've seen that 50% of them, 26 out of 52, were chromosomally euploid, and around 6% were mosaic. So up to the moment, we performed a single embryo transfer in 15 patients, and 8 of these patients got pregnant. And in one of them, we repeated the single embryo transfer. And we have a total of 8 embryos that implanted out of the 16 embryos that were transferred, and we got 1 miscarriage. So the results that we have as a summary are promising because from attending the type of patients that we are dealing with, that were patients that basically had not had blastocysts in their all previous attempts. So from the 25 that we did, we got at least 1 good quality blastocyst in 21 patients. And unfortunately, in 5 of them, all blastocysts were aneuploid. So we end up having only 16 patients that had at least 1 good quality embryo that was euploid, 1 good quality blastocyst that was euploid. And only in 4 patients we were not able to achieve fertilized oocytes or blastocyst development. So all pregnancies that were established were monitored very closely with regular scans, amniocentesis, and NIPT. And 5 of these patients have already delivered a healthy child. I have to say also that here, because it was the first embryo transfers that we were doing, we followed a very conservative approach, meaning that we transferred 1 embryo only when we were sure that everything was okay with the previous pregnancy. So in the second embryo transfer, we waited for having results and for being sure that the first embryo transfer was okay. So we waited more than 6 months to make sure that the first pregnancy was okay. And the same happened in the following embryo transfers. So in these babies and in the biopsied samples from the blastocysts, we have used a SNP analysis and DNA fingerprint to confirm the origin of the nuclear genome and of the mitochondrial DNA, which was confirmed in all the children born. And these children are being following a program that was established with a pediatrics hospital that mainly comprises neurological examinations according to the Hammersmith Infant Neurological Score, and also psychotherapist examinations, and also assessment of cognitive potential. So this is the plan that has been established to follow up the babies until adult age. Part of that, all the results that we've been collecting are being registered in this registry that we have created just for this study in order to record all results and track the children, because it's very important that we follow and we get all this information. So based on the evidence that we have so far from the research that we did, and also other groups that have been working with maternal spindle transfer, I think we can say that maternal spindle transfer does not adversely affect the spindle apparatus or early embryo development, but of course all the steps of the protocol and the culture system need to be very well optimized. Actually, if this is performed in oocytes with compromised quality, we are able to enhance their potential to develop up to blastocyst stage without impairing euploidy rates and with very low heteroplasmy levels. From the results of our pilot trial, we can also say that the embryos derived from my MST are able to implant and sustain a pregnancy of a healthy baby to term. Of course, we need more carefully controlled pilot trials to have more insights into the feasibility and reproducibility of the technique for clinical applications. I also believe that the results that we can get from this pilot trial may also be useful to have more insights on whether this technique can be useful to reduce the risk of transmission mitochondrial diseases. And finally, because we also think a lot about patients and we tend to forget about the donors, but this technique could be also advantages for the donors, because the children will not be genetically related to them. They will share only the mitochondrial DNA. And therefore, this could be or could reduce their psychological or anonymity concerns that sometimes are associated with conventional oocyte donation programs. Finally, I'd like to thank all team that has been involved in this project. Of course, Dr. Gloria Calderon, that was the mentor and had this idea in her mind since so many years, since the time she used it to work with the group of Dr. Cohen. Also, like to thank all our team in Embryo Tools, the group of Dr. Dagan Wells in Oxford for doing the all genetic analysis, and of course the group in Greece led by Dr. Psassas, Dr. Constantinos Kostaras, and all the team in the lab, Eros, Christy, Vanya, all of them. Thank you so much for supporting us on doing this project, and thank you also for the audience. And finally, just before finishing, I was reading not too long ago some articles from Professor Eli Adashi, and I found this sentence that I think is really interesting to share with you all, because it is something that we sometimes have in mind, but we have never had the opportunity to express it. And it's very, very true, I think. Thank you so much.
Thank you, Nuno. Dr. Costa-Borges, the very impressive presentation and the technical difficulty is quite impressive. One thing I'm wondering about is you did do PGT. What about mosaicism? I wonder if having a better spindle would lead to lower rates of mosaicism. Of course, none of us can totally agree about mosaicism either, but let's say that it does exist. Have you looked at that, mosaicism rates?
Yeah, this is a very interesting question, because it is true that mosaicism usually happens after fertilization, so during the mitotic divisions. So it is very likely that having a better cytoplasm environment, we could reduce the levels of mosaicism. And the results that we have now, and I showed it in this graph, is that from all embryos that were biopsied and analyzed by NGS, we have rates of around 5%, 6% of mosaicism, which I think could indicate something that indeed we are able to reduce the level of mosaicism, having a better cytoplasmic machinery. Of course, the numbers are still low, but this is something that we need to, that is definitely very interesting, and need to look into.
Yeah, that's fascinating.
Yes, thanks.
That was great. I mean, it's fascinating. We now have 15 babies, right? The two of you have 15 babies together. That is an amazing thing. I was just wondering, listening to the two of you,
I have
a lot of questions that come up, and one of them that is intriguing is the issue that Ailey earlier on raised, which is that of carryover, and so we don't see much of that happening at all in Nuno's work, and yet we see it in 2 patients. I think it was 2 patients or 3 patients in Pavlo's work. Of course, it is at different stages of the zygote. It could have just been, I mean, in both cases, I don't think it makes much difference, although Nuno may disagree. But I don't think, I mean, this is not a mutation-based heteroplasmy. This is a benign form of heteroplasmy, if you will. That's basically the hypervariable area that is different between these populations of mitochondria. But it is interesting because of the conclusions that Pavlo drew was, well, you don't do this in mitochondrial disease, because look what could happen, right? You clean up those 2 pronuclei, you have almost removed all the cytoplasm. It looked completely empty. What I saw on those pictures, it looked completely empty. In Nuno's case, that spindle is very small. You think there's a difference between the spindle in a way it sticks to mitochondria in terms of the difference with a pronuclear envelope, which has a membrane? Do you think that is a difference, that the pronuclear envelope actually is more sticky for mitochondria? Or do you think it has to do with the cell cycle? Because in interface pronuclei, the mitochondria may be more condensed to the nuclei. I certainly see that in somatic cells. Whereas in the metaphase, the mitochondria move throughout the entire egg. And is that maybe the difference that we see here? In other words, Pavlo, you still transferred 1 or 2 or 3 mitochondria, and in Nuno's case, it was 0. Or I mean, I'm probably exaggerating, but it was very, very, very small. And that may be what you're seeing here. Is that a possibility?
Pavlo, do you want to go first?
Well, why not? Okay, you convinced me. We'll move to polar body genome transfer. Here is believed that they do not contain any mitochondria as well. But we'll test it. We know that around pronuclei there are a bunch of mitochondria, and it's very hard to get rid of all of them. But as well as to spindle, in the very spindle itself, there is no mitochondria, but around it, the spindle needs energy. So there are some mitochondria, and it depends on the technique, I believe, not the cell stage, but still, I agree completely that when we perform the nuclear stages as GV transfer or PNT, we'll have more mitochondria than we perform spindle or polar body transfer. Probably, I think we need to ask specialists about it.
I think it's. it's described, and we have done this analysis in our lab as well with the Mitotracker, and it's indeed true that around the perinuclear area in pronuclei and in GV, for example, there is an accumulation of mitochondria that is not present around the meiotic spindle. So this could be one of the reasons for maternal spindle transfer being associated with lower heteroplasmy levels. And another reason is maybe something that we discovered by chance in the mouse work. That is, when we do the spindle transfer around the mitochondria that eventually are co-transferred with the spindle, remain around the spindle for the first hours, right? So when we perform the ICSI and the oocyte extrudes the second polar body, most of the mitochondria that are still around the spindle that have been transferred are expelled in the second polar body. So somehow it's cleaning a little bit the mitochondria that we have transferred with the spindle, and this might also contribute for the reduced levels of heteroplasmy in our case.
Right, right.
But Jacques,
Now,
that doesn't matter as much, does it? Because we're
Exactly,
treating embryo development as opposed to mitochondrial disease.
well, that's certainly true, but we're trying to learn, we're trying to see if there are lessons here for this very important technique of trying to do MRT, or cytoplasmic donation for mitochondrial disease, which is just a potentially beautiful application, but we don't want to do it wrong. So, so I think this is a good discussion, and it's kind of a reversed world now where you two and probably more have shown that this can be done, apparently with good success and apparently getting normal babies. But there is a lesson here for MRT for mitochondrial disease. What comes up from all of you is the polar bodies are mentioned all the time. And so this idea, and Ellie mentioned it first, this idea of using a polar body to do this technique rather than spindle and pronuclei. And the mouse work comes up, and it is indeed quite easy to do in the mouse work, provided you use the right strain. Because if you use an inbred strain, an outbred strain or an F1 hybrid in the mouse, that polar body just falls apart, as they do often, not always, but often in human patients. So if you are bound to do the polar body method, I would think that is very, very, very tricky, because the polar body is not the best cell in the world. It's a cell that has been discarded in a way. The oocyte is kicking it out. And if you wait an hour or two because of all the work that you need to do, are you not afraid, everybody here on this panel, are you not afraid that can cause problems, that that you are incorporating some cytoplasm of a cell that has been trying to, it's been basically discarded from the oocyte, or in the case of a second polar body from the zygote? What are your thoughts about this, everybody? What do you think?
Oh, I can answer to it if you won't mind.
Sure.
Okay, so let's not forget about the first baby ever born. I mean the Dr. Zhang baby in 2016. I'm in regard to mitochondrial transfer. I mean the percentage level of maternal mitochondria was as high as, I believe, 9%, even though it was spindle transfer, right? So it could be different between techniques, between labs, and so on. As for polar body, I know for sure that we have a baby born in Ukraine after polar body 1 genome transfer. So at least for one case, it worked. I'm pretty sure that it's fine. It depends on the size of polar body itself, because yes, they are designated to die, but I don't think that there are a lot of factors that promote this apoptosis. I think they die because of energy depletion or other stuff because they don't have all the machinery. But if you have the huge polar body, it happens time to time. You will see the formation of a nucleus inside this polar body, and this cell feels very good. So the problem is in the size of polar body. And if you use the fresh polar bodies, there should be no problems because DNA in it's quite good. I've done polar bodies a lot, and for younger patients, they work well, pretty well. I've observed only once the apoptosis process, but I believe that it was very old polar body, not the patient, not the cell, but the polar body. It was like many hours after fertilization. But for a fresh one, you can use them with no caution, I guess.
Okay. Nuno, do you have experience doing any polar body work?
No, not clinically. I guess it's an interesting thing that should be studied in detail. I know the work of Dr. Metalipov on this, and it seems to be useful to maybe also take more advantage, especially in cases of a very low number of oocytes. So we could duplicate the number of oocytes in patients with low response.
So, Ellie, if you could unmute, I think you're on mute.
Thank you. Yes.
There you go.
Just to say that we have recently reviewed the literature on polar body transfer, and the information, as you know, is now applicable to mice, to rhesus macaques, and to humans. And on the whole, we were left with the impression that polar body transfer seemed to be, based on what we read, with less in the way of carryover. Because you begin with a structure that is cytoplasm poor in relative terms, and therefore mitochondria poor. And the literature that is out there right now on all these three species, I think seems to bear that out. So that would be one point I would make. The other, since I'm surrounded by embryologists and I'm obviously at a disadvantage when it comes to the technology, but there is interesting literature that we've begun to think about that deals with the circumstances under which the transfer is made. For example, mitochondria tend to be perinuclear, but that is temperature dependent, at least in part, as one example. And so it could well be that physical and other manipulations could be used during the transfer procedure to minimize the likelihood of mitochondria transfer, temperature being just one of them. So just looking at the literature of very basic mitochondrial science, localization of mitochondria at certain circumstances could well also, I think, improve the ability to transfer nuclei without or with as little as perhaps no mitochondria, if that's ever possible. So I think there are some clues to improving carryover. Polar body may be one approach, potentially, if it's verified, and rethinking the physical circumstances under which the transfer is done, parameters such as temperature and others potentially influencing where the mitochondria are in the cell at the time of the transfer. And if you can get the mitochondria, for example, to be peripheralized, and then you remove the nucleus, the odds are you'll have very few mitochondria carried over. So there are some, I think, thoughts there, concepts there that are at least worth pursuing. And of course, only people like yourself who are actually doing it can potentially explore it, ideally in a mouse, of course, first. And if that works, then potentially apply it elsewhere.
Yeah, no, it's very good. These are very good points. Another topic is related to, again, to carryover, and you're suggesting earlier that the CRISPR technology or genetic manipulations so that mitochondria are basically becoming, well, either destroyed basically and don't have a mitochondrial DNA anymore, or not a functional mitochondrial DNA anymore, which has been done in the mouse using different technologies, but talents in particularly, which is a really elegant work that was done years ago. And I just think, could you combine, I mean, it's maybe crazy, okay, but could you combine what Nuno is doing and what Pavlo is doing with your suggestions, and doing that on the pronuclear karyoplast or the spindle cytoplast, for lack of a better word, but that's of course also a karyoplast. Could you combine that? Because they seem to be quite viable. I don't actually know if you can freeze these karyoplasts you make, Pavlo, but could you use, so that means that they will survive for hours. Could you use your suggestion, Ellie, on those rather than the entire cell?
Did you mean editing?
So could you use talents, for instance, on a karyoplast or a spindle karyoplast or a pronuclear karyoplast, rather than using talents or CRISPR to work on mutated mitochondria and sort those out? And
I think that remains to be seen, but in a review that we just, or perspective it's called, that we just submitted to Science, I don't know how well it will do. The last paragraph reads, transfer plus, replace and edit, question mark.
Okay.
Not replace or edit.
Okay.
Replace and edit, question mark. It's a short paragraph because it's just an idea.
Sure. Yeah.
But if you combine the power of these two technologies, potentially, as I think is what you're suggesting, we might be in a better place. That said, I think, as I pointed out, as exciting as the new editing option is, it's at this point highly limited and suffers from major shortcomings in the context that we work in reproduction. So I don't want to make too optimistic predictions. I don't see it replacing, I don't see it replacing replacement technology tomorrow. I think it has a future potentially, but I think much remains to be done. As you know, gene editing, even for other purposes in the embryo, is nowhere near ready for prime time. So that technology is lagging at this point. Will take a while to improve it. And so I think we are going to continue to have to work on replacement technology. And some of the ideas I mentioned, namely work with polar bodies potentially, change the physical parameters of the technology while we're doing it, those could be game changers. They may not give us 100% or zero carryover, shall we say, but it will be an improvement potentially. That's where I would focus right now. I think the editing is very interesting, very promising, but nobody is saying that it's anywhere near ready to assist us in this context. We need many more base editors, first of all. This one can only do one set of base pairs. We need several options before we can actually make this work. So we're in transition, like every piece of science. It progresses in small steps, and then all of a sudden we wake up and looks like we came somewhere, we arrived somewhere. Time will tell.
Jacques, I find the this idea of creating more eggs using the pronuclei very, very compelling, because I think that seems a lot closer to reality and clinical use than this idea of skin cells and converting skin cells back to primordial cells and creating stem cells And obviously, embryonic stem cells have so many barriers as well. So it seems like just increasing the number of eggs available using this pronuclear technology might be
Polar body.
something
Polar
that would
body.
be
Polar body.
polar body technology might be something that could be clinically useful way before you know, the stem cell use or somatic stem cell
Yeah.
creation of gametes, which obviously is one of the hugest barriers we currently face with egg, you know, maternal or female infertility or egg-based infertility.
Yeah.
So who's doing that? Are you guys going to work on that soon? Because we need some more eggs.
Well, we have a clinic in Ukraine who perform this procedure. They just they make their advertisements as they are multiplying the eggs. So this is clinically applied now.
So they have
At least in Ukraine. Yeah, they have one baby reported, but only in Ukraine they have no publications as far as I know. But I wanted to mention that as far as I understood, Nuno worked only with the frozen patients' oocytes. That's, I believe, diminished the number of mitochondria very well. And we prefer to do the same. We ask patients to transfer or send us their oocytes in a frozen condition. And then we thaw them, do all the manipulations, and that's another way for reducing the mtDNA.
Do you think that the freezing and the thawing and the loss of the mitochondria is responsible for the lag in blast development in previously frozen oocytes?
No, I don't think so. I think that it could be helpful for nuclear transplantations, but when you say about an embryo, it differs because at the blastocyst stage you have only a small number of mitochondria in each blastome or in each cell of the blastocyst, because there are huge extinction of mitochondria after genome activation, and only the fittest will survive. So at the end of the blastulation process, you will have only few mitochondria in each cell. So I'm not sure that they're responsible for this lagging. I think that it could be helpful for nuclear transplantation, but we have very good results on frozen oocytes and frozen embryos, and we have millions of kids around the planet. So why should you say
So that
it's...
hasn't really been a barrier. The making blasts from frozen eggs has not created a barrier for you. It hasn't
Of
really
course not, no.
dramatically reduced the blastulation rates.
No.
Okay.
We have not observed anything like this.
Talking about success rate at different stages, what about fertilization rate? Of course, that doesn't apply to pronuclear transplantation, but to MST. I mean, there was a recent paper published just a few months ago from a Czech group, and they apparently done cytoplasmic transplantation for many years and published retrospectively their results. And just looking at and I see a question here from the audience. There's a series of cytoplasmic transfer cases that showed an improvement of fertilization. Is that something you have seen, Nuno? Did you see an improvement of fertilization?
Well, in some patients that had also problems in fertilization, their oocytes, we've seen that with spindle transfer we could normalize these rates. So I guess, yeah, it's around 75–80%.
Okay.
Of the reconstructed oocytes. So
that's fascinating.
I guess this is normal. I think, in general terms, what we're seeing is that doing the maternal spindle transfer, we have results similar to an oocyte donation program in terms of fertilization, blastocyst formation rates, with the advantage that we're dealing with these patients that have several previous attempts with poor fertilization or poor embryo development afterwards. So this is, I think it's the technique that I've been working with, where we see the greatest improvement so clearly. So yeah.
Great, great. Thank you.
I think, sorry, I think Nuno has a point here, because it's, well, we need to highlight this. It's very patient or case dependent.
Okay.
Yeah, of course, it's meant only for patients that we're sure that have a poor embryo development due to poor site quality, of course.
Yeah.
Or poor fertilization.
Yeah, exactly.
Any suggestions? You know, what we're really missing is an assay, right? A diagnostic assay. I think the mitochondria experts have argued that, say, well, we know about mitochondrial disease and we know there are these mutations, and sometimes they're very high and sometimes they're most 9 out of 10, 8 out of 10 cases higher than what's considered safe for transfer, particularly when you do PGD. So on your side, on this infertility debate and doing these manipulations, what is the assay? Are we going to have a diagnostic assay? So you can say, well, this patient, I think Pavlo already indicated top 8 as one little breakthrough there, where you have a gene that is involved in cytoplasmic maturity, cytoplasmic competence. Can we develop an assay? Is that a possibility? Yeah, Nuno, you're nodding. Is that a possibility to?
I think it's the way the genetics field is evolving, and having many possibilities of screening the patients for different genes. We will end up, I believe, in a near future to identify the real causes for this defective cytoplasm.
I think there's a lot of panels available now. It would be interesting to test your patients now. That
this is actually a project that we are working on, and
cool.
So we would like to identify the real causes. Of course, the problem that we have now is similar to what happens nowadays with oocyte donation programs, when these patients that don't have other alternative than going to oocyte donation. So we are dealing with the same decision when we have to indicate whether these patients want to go to spindle transfer or to oocyte donation.
Thank you. I really hate to stop this, but we have broken a record. We have broken a record. We are exactly 24 minutes past the longest session we've had before. So I unfortunately do have to stop this, and this is very unfortunate because the audience was great and a lot of great questions, and we could continue this, I think, for many hours. The 5
We are the champions here.
Yeah.
Our mitochondria are working hard.
Okay, so next time we'll have to get back to this subject. I really think in a year, and I think we have to get back certainly to the mitochondrial approach and the mitochondrial disease people with Adashi, hopefully in the spring. So that is the plan, and I think all of you should come back to talk more, and hopefully new people to talk more about the prospects of this in infertility treatment. So thank you everybody for taking time to participate and prepare this session. I particularly like to thank you, the 4 speakers, and Serena for also being a moderator. I like to emphasize again that this session was generously kindly supported by both Embryo Tools from Barcelona, as well as the Nadia Clinic in Kiev in the Ukraine. And I like to thank the organizing committee of this initiative, Peter Nash, Thomas Elliott, Shaista Sedrudin, Marianne Savetic, Giles Palmer, and their continued efforts. This is a lot of work. You're doing this very well, and it's really wonderful to work with you. Also, one of the more complicated elements of these webinars is the stream of questions that comes in. So I'd like to thank the triage team, which are all the persons I just mentioned, plus Anna McLaughlin, and today it was also Kelly Catterton. Thank you both for helping out. So this session will be available within about 24, 48 hours online, and you can watch yourself again, and of course, if you're in the audience or you have friends, please tell them to do so. And
Okay,
usually,
one more picture.
yeah,
One more
picture. Go
One
ahead.
more picture.
Well,
Everybody
I keep talking,
smile.
okay?
Okay. Okay.
We thank Thomas Elliott for his superb technical skills in arranging these sessions, and Giles Palmer for being the key producer. Thank you, Giles. So this has been an extraordinary year. We started this in the end of March. We have lost so much, and yet we gained also so much more in a different way. So our year here at I3 is coming to an end almost. We're not there yet. Tune in next Tuesday for a special and final webinar with a different format sponsored by I3. Please join us, same time, same place, next week, Tuesday, December 15, the 15th of December. The session is called Ethics in IVF, a networking event. So first there will be a relatively short keynote presentation by our own Kimball Pomeroy, touching on the broad topic of ethics, IVF, and embryology. And then the audience, that's you, will have an opportunity to discuss and debate your thoughts and opinions out on this subject out loud. I know in this formulation that we have this webinar running, you have to ask us the question and then we pose it to the speakers. Next week, you're in charge. So please come and join us because you will be guided into icebreaker rooms afterwards in about 15 rooms, I think. We have a maximum possibility for 300 attendees. Please check that in and hopefully everybody can get into those rooms. So please follow that information on our website and our social platforms for updates. And thank you again. Thank you, our audience. Thank you, speakers. And Serena, you're the wonderful moderator. So see you all next week, hopefully. I'd like to end up with how Adashi always signs his emails, which is please stay safe and stay in touch. All right. So see you next week. Take care.
Thank you. Bye.
Thank you.